Good evening, everyone. Welcome to Botany Insider's new free class. We'll be discussing about further topics in this particular segment. Please let me know in the chat section whether the audio and the video is clear to you or not. Good evening to all the students. Just confirm whether the audio and the video is clear or not. Good evening, everyone. Okay, so let's begin with our class now. Others would join in the time being, right? First of all, welcome to Botany Insider's class. If you're new to Botany Insider, please make sure to hit the subscribe button. Also, download our application. We have a number of courses running for you all. We have a full course, we have a crash course, we have your notes booklet. You can order your notes booklet from via WhatsApp as well and via our application as well, available both on the iOS as well as on the Android application. Okay, so for the notes booklet, you can WhatsApp us on this particular number. You'll get all the details and special discount as well. Okay, so download the app and get yourself enrolled. In addition, we are about to start a PYQ course for the December 2023 examination, right? So let's begin with our class now. In this class, we'll be discussing about the major segment. If you remember, yesterday we discussed about GPCR. I'm expecting all the topics that we have discussed yesterday along with the previous question is clear to you. Today also we'll be discussing one more important segment which is related to your IP3-DAG pathway. Okay, it is again one of the most important segment in the CSIR. It has been asked repeatedly. I'll be showcasing two questions at the end of the class in the same manner we discussed yesterday. Today also, first we'll understand the entire process and then we'll be solving out the question also. Okay, so let's discuss about the IP3-DAG pathway. Okay, so in the IP3-DAG pathway, first thing to understand is the type of the receptor utilized over here. The receptor used over here is your GPCR itself. Okay, what is the functionality or what are the various functionalities of your entire GPCR? Let's understand. Let's recall that particular thing. Okay. Yesterday we had a discussion. What is GPCR all about? It is G protein coupled receptor. There's a plasma membrane. On the plasma membrane, you have a serpentine receptor, right? The N terminal, C terminal, right? In addition to that, as and when the ligand binds to your N terminal, information is transmitted to your G protein. Talking about the G protein, so G protein is having three of the subunits, alpha, beta, and gamma, right? In the alpha, beta, and gamma, you had this particular orientation. Then the alpha subunit gets activated as and when your ligand binds to it. How does it get activated? You all also participate in the class. If you remember any of the fact that we discussed yesterday, you can keep on writing down in the chat section. Okay, it will help you understand or it will help you revise the facts better. Okay, so you have the G alpha subunit getting activated. How? With the help of guanine exchange factor. We had a discussion. This guanine exchange factor is converted how? Via GDP getting converted into your GTP, right? GDP getting converted into your GTP with the help of which, what do you have? With the help of which you have your G protein getting activated with the help of your, like now it is bounded with your GTP. Okay, till this particular point, the segment is similar. Okay, till this point, till the time your G protein with the alpha subunit getting activated, it's clear. It's the same what happens in the previous one. Now comes the change in our GPCR, in our IP3-DAG pathway. How does it actually operates? Understand this. Very, very important. Okay, understand this entire segment now. What happens? G alpha, your G protein with the alpha subunit is activated. What it will be doing? It will be interacting with your PLC beta. What is PLC? PLC stands for your phospholipase. I'll be showcasing all of these terminologies. Wait for it. PLC stands for your phospholipase. Now what happens? This phospholipase getting activated will help in the conversion of your PIP2 into your IP3 and your DAG. Okay. PIP2 gets broken down into IP3 plus DAG. What is DAG? DAG stands for your diacylglycerol. DAG. It stands for diacylglycerol. Okay. Now, your IP3 is what? IP3 is what? IP3 is your phosphatidyl triphosphate, right? So, what is it? Sorry, it is inositol triphosphate. What is IP3? It is inositol triphosphate. And at which particular position do we have the phosphates being present? Yes. Any idea? IP3. In IP3, at which particular positions do we have the phosphates present? This has been asked in the examination. It is first position, fourth position, and fifth position. So, inositol 1,4,5 triphosphate. This is what your IP3 is all about, right? Now, what happens? IP3 will be leaving the plasma membrane. This complete process is occurring in your plasma membrane itself. Okay. Your DAG is also attached to the plasma membrane, IP3 also, PLC beta, all these things are occurring in the plasma membrane itself. Now, what do we have? Now, your IP3 will dissociate from the plasma membrane and it will enter, or it will be going towards which particular organelle? It will be going towards your smooth endoplasmic reticulum. Where it will be going? It will be going towards your smooth endoplasmic reticulum, right? In the smooth endoplasmic reticulum, what do we have? In the smooth endoplasmic reticulum, you have ligand gated ion channels. Okay, what do we have? We have ligand gated ion channels, right? Inside the smooth endoplasmic reticulum, you have calcium being here. Okay, over here you have ligand gated ion channels which gets opened as a result of IP3. How does it get opened? It gets opened as a result of binding of IP3, which leads to the activation of your channel, which leads to the activation of your ion channels. That is, your calcium comes out of the Ca2 plus and it will be moving towards your plasma membrane region. As and when it moves towards the plasma membrane region, it will be forming a complex with your DAG, which leads to all together, they lead to the activation of PKC. What is PKC? PKC stands for your protein kinase C. Protein kinase C will be entering into the nucleus. In the nucleus, it will be opening, or it will be starting off, or initiating, or it will be activating a number of transcription factors. As and when the transcription factors get activated, you get to see the final response. Very important. Repeating it again. Calcium 2 plus is released as a result of the opening of the ion channels, because your IP3, your inositol triphosphate, gets attached to the ligand gated ion channels. Ca2 plus gets released. Ca2 plus interacts with DAG in your plasma membrane, where both of them helps in the activation of PKC, protein kinase C. Then you have the entry of the PKC into the nucleus, and finally the response is seen depending upon what ligand was there. Okay, so this is how your complete GPCR actually operates. Very, very important. The initial thing over here is something that you understood yesterday. Now in the IP3, there are additional steps. From every step possible, the question can be formed. Okay. In addition to that, over here at this particular point, at this particular position, there is utilization of Kelmodulin. Okay. Kelmodulin also gets utilized over here, which helps or which works as a carrier for transferring the Ca2 plus from your cytoplasm or cytosol towards your plasma membrane region. So this is how the entire IP3-DAG pathway operates. Okay. Quickly go through this. If you find any difficulty, any problem, you can ask. Otherwise, I'll explain you the further pointers. Okay. Clear till this particular pointer, everyone? Just quickly go through this. If you find any difficulty at any point, you can ask. Okay. I'll be showcasing a number of questions just after this. Like when we get through the theory, we'll be solving out the questions also, and you'll understand that from which particular points questions are there. Okay. Just let me know in the chat section whether till here everything is clear or not. Quickly. Clear? Very good. Okay. Now let me show you the theory part for the same. See over here. IP3-DAG pathway. What is the other name for the same? The other name for the same is inositol triphosphate diacyl glycerol GPCR pathway, right? The ligands for the same are, the ligands could be hormones, they could be neurotransmitters, they could be growth factors, hypertrophic stimuli such as angiostensin 2. Now, PIP2 stands for what? It stands for phosphatidyl inositol diphosphate, the phospholipid which is present on the plasma membrane. Then you have PLC, which is phospholipase C. DAG stands for diacyl glycerol. It is hydrophobic. Remember this. Star mark this. I'll be showcasing you a question. Okay, it's hydrophobic and physiological activator of PKC. It has high affinity for Ca2 plus, that is calcium 2 plus. Now PLC beta, it breaks down or it breaks PIP2 into DAG and IP3. DAG remains on the plasma membrane and IP3 diffuses to the cytosol to enter into the ER. It does not actually enters in it. It is binding to your outer membrane, outer ligand gated channel so that the Ca2 plus can be removed out, right? PKC, it is a kinase acting on the serine threonine residues that translocates from the cytosol to the plasma membrane, that is utilized in the formation of your DAG, which is your diacyl glycerol. Very important, right? Now look over here. These are my notes, like what I made during my preparation time. Understand this. IP3 is inositol 1,4,5 triphosphate. This is your GPCR, G protein, like the external site where your ligand comes and joins over here. Then you have the 7 serpentine, like the 7 times membrane spanning through the serpentine. G protein gets activated with the formation or with making a complex with the GTP. Beta gamma stays. Alpha will be interacting with PLC beta. After interacting with the PLC beta, what is there? You have PLC beta helping in the breakdown. See, PLC beta catalyzes the reaction where the PIP2 breaks into DAG and IP3. DAG remains in the nuclear in the plasma membrane so that you have the so that it can stay over there because it is having hydrophobic property. IP3 diffuses with the cytosol and enters the ER, or it can say it stays on the outer membrane. Then you have Ca2 plus coming out, it binds to Kelmodulin. After binding to the Kelmodulin, it will be making a complex here. PKC gets activated and finally the response is seen after it enters into the nucleus, right? I've explained you the entire same thing, right? Exact same thing I've explained you just now. This is about your IP3-DAG pathway, okay? Now, over here, you have the significant importance: calcium signaling, protein kinase activation, cell signaling and communication, integration, and disease relevance as well. Okay. Now, if I tell you about Kelmodulin, remember about Kelmodulin that it is what? It's a calcium binding protein that plays a very important role in number of cellular processes. For example, the processes which requires calcium requirement, right? So talking about the structure, so look into the diagram. This is how the calcium Kelmodulin looks like, made up of almost 148 amino acids. Over here, what do you have? You have two globular domains connected with the help of a flexible linker. So one domain, second domain, the flexible linker. So this is how a Kelmodulin actually looks like, right? Then you have calcium binding and the conformational changes, target protein regulation, and finally the response, the cellular functionality. If any particular cell is not able to take up the cell properly, take up the calcium properly, so this Kelmodulin helps in this particular process. Okay, so this is how that's how it's actually done. Okay, so this is about your Kelmodulin. Okay. I hope it's clear. And with this, we have the major segment done, right? Get or read this particular question, everyone. Try finding the answer. Yes, Nisha, I have tried to make the notes as simple as possible. Okay. And you're an enrolled student. You know how we are doing the things in the classes, right? And same Lena also. She just joined today. So you can understand how important these notes are. With just this particular thing, like, see, these notes help you get through the entire thing within a minute. During the examination, we cannot go through the theory, right? So, at this time, when we have the exam round the corner, these notes, like I have added these exact notes, these exact things in my hard copy notes as well. So, it's very important so that you can revise the things very, very easily. Okay. So, get through this particular question, everyone. It's a very important question. This one is June 2021. Easy question. Localized increase in the cytosolic level of your free calcium. They are critical to the function as secondary messenger, right? Calmodulin, which is a small cytosolic protein, mediates many cellular effects of the Ca2 plus. So some information is given to you. We need to find out which of the following is not correct. If you had understood or if you had paid attention to the entire class, you will not get it wrong. Okay, take your time, mark the answer for the question, everyone. Give it a try to this question. 15 of you are there in the class. Mark the answer for this, everyone. Quickly mark the answer, everyone. Very simple question. Only two of you have marked the answer till now. Very good, very good. Read the question carefully. You'll get the answer very easily. Okay. Excellent. Four of you have already marked it. Others also try marking the answer. First option is each Kelmodulin binds to six of the calcium ions in the cooperative fashion. Then binding of Ca2 plus causes the Kelmodulin to undergo a conformational change leading to the active Kelmodulin. Then since the binding of Ca2 plus is cooperative, a small change in the level of the cytosolic Ca2 plus can lead to a large change in the level of activated Ca2 Kelmodulin. And last, one of the many enzymes activated by the Ca2 plus is CAMP phosphodiesterase, which degrades CAMP and links the calcium 2 plus and CAMP signaling. Very good, very good. Answer for this question is yes, option number 1. You can understand, like, just by getting through the main concept, you can mark the answer. This one is June 2021. Easy question. It was a 2-marker. And if you remember just one fact that Kelmodulin binds to 4 of the Ca2 plus and not 6, you can easily reach to the answer. So 6 is incorrect. It binds to 4 of them. Look into this. I've just explained you. 1, 2, 3, and 4. It is not 6. Okay, very good. Many of you got the answer correct. Over here as well, it's written. So each domain, there are 2 domains in total, and each domain binds to 2 calcium. So in total, 4 Ca2 plus are there. Remember about this particular thing, right? Answer is option number A. One more question for you all. Give it a try to this question, everyone. Very easy. In the context of signaling, the protein kinase C depends upon multiple protein molecules in its complete activation. So the question is, this activation depends upon the presence of which of the following. Now over here, you'll have to apply your basic knowledge as well. Give it a try to the question, everyone. And because you need to find out combinations. So try eliminating as in when you get one of the option incorrect. Okay. Try it out, everyone. Very easy. Activation of PKC depends upon which of the following. Very easy question. Understand this? First option is phosphatidylserine on the inner leaflet. Second, calcium 2 plus. Third, phosphatidyl ethanolamine on the inner leaflet. And fourth is your diacyl glycerol on the inner leaflet. If you know the basics of your plasma membrane also, you can easily mark the answer. Take your time, mark the answer for the question, everyone. Very good, very good. Everyone participate in the class. Excellent. Let's look into the answer now. Very easy. See, calcium will for sure be there in the options, right? Because our PKC activation depends upon it. So B will always be there. So B, B and B. I can eliminate second one. Then diacyl glycerol, DAG present on the inner leaflet. Again, correct. We have just understood this in the IP3-DAG pathway. So D should also be there. So either the answer is B, either the answer is 3 or either the answer is 4. And if you look into the answer key, the answer is option number 4. Phosphatidyl serine on the inner leaflet is correct. Ethanolamine is not on the inner leaflet. It is on the outer leaflet. Okay. So C option is incorrect. A option is correct. You can reach to the correct answer with just your basic knowledge. If you know the basic information regarding your plasma membranes, distribution of phospholipids, and the concept of your IP3. See, over here, they have nowhere written that this question is related to IP3-DAG pathway, but you need to have a knowledge of this pathway for solving out the question. Are you understanding this, everyone? 7 to 8 of you marked the answer and all of you got it correct. I am expecting that you know the basic concept of plasma membrane. And because it's almost end of August, exam is in December, so you should get done with your syllabus very soon, and then you should focus upon revisions. Clear, everyone, till this particular pointer? Yes or no? Right, so this is about it. Now, just let me know in the chat section which other topics you want me to take. From tomorrow, Khushi ma'am, like we have one more educator. She'll be taking 3 classes for your unit number 13 now. Okay. She'll be taking 3 classes for unit number 13. Be there in the class. She'll be taking the methods in biology in the best manner. Okay. And tomorrow will be a very, very important class. She'll be taking the all the techniques basics. Okay, she'll be explaining you the basic principle of each and every technique, okay, in 30 minutes. So it will be a rapid 30-minute session. Be there in the class, okay. Tomorrow, like for the next 3 days, she'll be taking the class. Now you can let me know in the chat section which particular signaling pathway would you want me to take in the next class. Whenever I'll take a class, like most probably on Sunday, I'll be taking. Just write down in the chat section, not from unit number 13, from any other unit also if you want. Write down which topic you want to study over here. And if you're looking for a very structured thing, then we have a crash course, okay, and a full course running. So if you want a very structured preparation with us, you can come in the crash course or the full course. This is the full course and this is the crash course running right now. And you can suggest any topic if you want, or if you're facing any problem in any of the topic, we can take up. Quickly let me know in the chat section, everyone. Any topic of your choice which you want to study here. Otherwise, we'll take the topics from our end, whichever we feel like the most important ones. Any suggestions? Cell cycle, okay. What else? Anything else? Okay, so you can suggest the topics in the comment section also. Those of you who will be watching the recordings. Nisha Balyan is saying cytoskeleton. Okay. I'll take up these topics. Okay. You can suggest down the topics in the chat section also after the class, and we'll be taking up the classes accordingly. And tomorrow, be there in the class. Kushi ma'am will be taking unit number 13, all the units, like all the techniques that are there in the most simplified way. How can you remember in the most simplified way? How can you get done with the entire thing? We'll be discussing. Okay. So we'll be getting the new class tomorrow. All the best. Good luck. Take care, everyone. Thank you for joining in. Let's meet in the next class. Okay. And do not forget to hit the subscribe button. We have to reach to a golden number by the end of this year, which we are targeting 20K. So let's reach 20K very, very soon. Right? Hit the subscribe button. Share the channel to as many friends as possible. Let them also know about it, about our channel, about our platform. Okay. Thank you for joining in. Bye, everyone. Take care.